Imagine the next big cancer breakthrough comes from something as simple as yeast. Does that sound impossible? Maybe think again. Model organisms, creatures like fruit flies, roundworms, and yes, even yeast, have been at the heart of some of the biggest scientific breakthroughs. These seemingly simple organisms give us deep insights into complex biological processes that apply to humans too. It turns out that even though these organisms are very different from us, many of their molecular and cellular processes are remarkably similar to ours. You see, life on Earth shares a common ancestor, meaning many of the basic building blocks like DNA, proteins, and cellular components are remarkably conserved across species. What I mean by conserved is that many of these processes have remained almost the same throughout all of evolution. Whether you're looking at a yeast cell or a human cell, the way they produce energy, replicate DNA, and divide follow the same basic blueprint. So when scientists study these processes in model organisms, they can uncover principles that apply to human biology as well. In other words, by studying these simple organisms, we get a window into the core machinery of biology. Machinery that, with only slight variations, drives life in us. That's why these simple organisms have been at the core of some of the biggest biological breakthroughs, and how they can hold the key to some of our biggest challenges in medicine. One of the benefits of using model organisms is that they have short generation times, meaning they reproduce really quickly, meaning that scientists can study many generations within a relatively short period of time. Imagine trying to study genetics in humans if it took decades to see how traits were passed down. E. coli can reproduce in a matter of minutes. Fruit flies, you can have generations just in the span of a few days. This speed gives researchers a huge advantage. And with that comes the fact that they're just easy to maintain in a lab. A few vials of flies or a tank of worms can provide researchers with thousands of subjects to study. This makes it easy to run experiments and generate massive amounts of data, all at a fraction of the time and cost it would take to use larger, more complex organisms. And these advantages, simplicity, rate of reproduction, and ease of use, are why scientists have been turning to these model organisms for over a century. They make a lot of experimentation accessible. Let's travel back to the early 1900s. Columbia University. There's a clutter of microscopes and fly bottles, and there stands Thomas Hunt Morgan. Morgan was not convinced by the idea at the time that traits were inherited by discrete units. Many biologists at the time believed in a more fluid inheritance, that traits sort of blended together like paint colors. So Morgan started studying fruit flies and meticulously tracked the inheritance of traits like eye color, wing shape, body size. Morgan observed that certain traits were always inherited together, which led him to propose the idea of linked genes. Genes that are located close to each other on the chromosome and would always be inherited as a group. So he and his lab started breeding crosses, generating and analyzing tons of data, and they were able to map the relative positions of genes on the chromosomes, which laid the foundations for future work known as genetic linkage mapping. Morgan's experiments showed that genes weren't just some abstract idea, they were physical entities on the chromosomes, which forever changed our understanding of inheritance. His work with Drosophila didn't just win him the Nobel Prize, it solidified fruit flies as one of the most important organisms in biology. And science continued to make extremely significant breakthroughs using model organisms. The discovery of programmed cell death, or apoptosis, that comes from work on nematodes, and it's significant in understanding development, neurodegenerative diseases, and cancer. Work with these C. elegans won the Nobel Prize in 2002. Now, it's important that you understand this because scientific funding is constantly being called into question. Probing questions of how to figure out and cure cancer might end up just looking like funding for fungus. Funding research into Alzheimer's might just look like funding for fruit flies. And when politicians at all levels try to cut out wasteful spending, they might end up cutting funding for these critical areas, either because they don't understand the significance of what they're talking about, or they're relying on your ignorance in not knowing what these organisms are for. And this is not just me trying to fearmonger. This actually was the case in 2008 on the national stage. During the presidential race of Barack Obama and John McCain, the conservative vice presidential nominee Sarah Palin was giving a speech. And in talking about wasteful spending, she said this.
Of reprioritizing. We've got a $3 trillion budget in this country, and Congress spends some $18 billion on earmarks for their political pet projects, and that right there is more than the shortfall to fully fund IDEA. Where does a lot of that earmark money end up anyway? You guys have heard some of the examples of where those dollars go. You've heard about the bridges, and you've heard about some of these pet projects that really don't make a whole lot of sense. And sometimes these dollars, they go to projects having little or nothing to do with the public good. Things like fruit fly research in Paris, France. I kid you not.
Shortly after, the journal Science published their response. Not only is this research just really cool, it has to be understood so that it can be protected. I'm Simon. If you like this video, give it a like and subscribe to keep talking science with me. Thanks.